This demonstration will show a typical workflow on the VersaProbe 3. In this example, we will be collecting XPS spectra from two insulating samples that we assume to be uniform films. During the analysis, we will use several automated features of the VersaProbe, such as the analysis queue, charge neutralization, auto Z-height, and SXI imaging. The SXI will provide a fast way to check our sample surface uniformity and allow us to confidently choose areas to analyze. We will be using our SmartSoft acquisition software. This package contains many features that allow us to collect spectra, depth profiles, line scans, and maps, and we can also control system options such as ion guns or photon sources. To start with, we can see a photo of the sample holder that was collected in the introduction chamber of the VersaProbe. This is a 60 millimeter diameter sample holder. There are two samples we will be looking at. The first is PMMA polymer, and the second one is an SiO2 on silicon. Both are insulators, and therefore we will use our dual beam charge neutralization system during analysis. Now to start with, we can choose where we want to do our analysis simply by clicking on the photo in the desired location. And so what I will do is click and select a point on the PMMA, and then I can click and select another point on the SiO2. And you can see this will populate the position list below, showing the stage coordinates of these two samples. I can then name each of the samples appropriately. Now that we have chosen the locations of interest, we can set up the acquisition parameters we want to use, such as the binding energy regions and the X-ray beam size. A variety of predefined beam sizes are available, or you can create your own that is appropriate for the size of your features of interest. The VersaProbe allows you to analyze both small features using a small X-ray beam size, or look at large uniform areas with our large area setting. Once you have chosen settings that you like, you can save those into a recipe. And so here we have loaded a recipe for a survey using our large area or high power analysis beam. This will allow us to collect a survey across a region of the sample that is approximately 1.4 millimeters in size. Next, we would choose settings that would help us for this analysis, such as charge neutralization if we have an insulator. In this case, we do have an insulator, and so we will use our dual beam charge compensation system that consists of electrons and ions. We can also choose other options, such as an auto Z or Z height alignment, and also we can collect an SXI photo, which I will explain later. This allows us to check the uniformity on the sample. Once I have the desired settings, I can choose to add these to our analysis queue. And so we can create a sequence of steps for the analysis. As you can see here, first I have chosen the PMMA and I added this to the queue. Then I chose the SiO2 and have added that to the queue. Each of these items has a number of tasks associated with it. For example, setting up the charge neutralization, doing the auto Z alignment, and collecting an SXI photo. To help keep track of my acquisitions, I can rename them. And so I'll rename the first one PMMA survey and the second one as SiO2 survey. And if I want to make sure that my queue is correct, I can validate the queue before I continue to make sure I don't have any errors. Once I'm satisfied with the queue, I can click the Go button, and this will start the analysis of the queue. So the first thing that will happen is the stage will move to the location for my analysis of PMMA. And the blue crosshair on the photo indicates where the stage is. And we will be able to see it moving as the stage moves to the analysis location. The stage coordinates are seen at the bottom of the screen, and the yellow box indicates that the stage is in motion. Once the stage coordinates of the analysis position are reached, the next step is a Z alignment. This process will move the stage up and down in the Z direction to maximize signal intensity through the analyzer. This makes it possible to load samples of different heights and easily set them at the optimum analytical position. Before we start the Z height alignment, we should turn on the electron and ion neutralizers. Because we had both of those set to auto, they will turn on as needed for the analysis. So there's no further user input needed. Now that the neutralizers are turned on, we will start the z-height alignment. As you can see in the plot, the stage is moving in the z direction up and down, as you can see from the x-axis of the figure, and we are monitoring the intensity as we move that height. Initially, we will take large steps, and once the intensity starts to increase, we will back it down and take a few smaller steps to find the optimum z-height. And as you can see, the intensity increased, reached the maximum, and we could see a decrease as we went up further. So we have found our maximum intensity at the proper z-height. After we have found the proper z-height, the next step is to take an SXI photo. SXI stands for X-ray Induced Secondary Electron Imaging, or scanning X-ray imaging. We use a sub-10 micron diameter X-ray beam to scan across the surface. At each point that is scanned in the 1.4 by 1.4 millimeter area, secondary electrons are produced. We can see contrast in the SXI image that might be representative of chemical changes. So this gives us a quick way to look for inhomogeneities in the analysis area. For our analysis, we'll be collecting a 1.4 millimeter area across the center of this image. As we can see, it looks fairly uniform, which gives us confidence that our analysis will be representative of the sample. Now that we have finished the SXI, the next step is to set up the survey. As before, the charge neutralization system automatically turns on and prepares us for the analysis. So as the spectrum collects, we can start to see a few peaks. PMMA is a polymer containing oxygen and carbon, so we would expect to see those on our surface. And we see a large peak at around 530, presumably oxygen, and carbon around 285 is what we would expect to see. We can use an auto peak ID to also identify those, and they also show up as carbon and oxygen as expected. So the next step that we may want to do is look at narrow scans of these regions to get more information about the chemical state. Since we had these transitions labeled on the spectrum, we can use an import function to automatically read those in to our settings. Since we're doing high resolution scans, we will use a lower pass energy to give us better energy resolution. And we can also choose the number of sweeps or cycles to adjust our analysis time. Since we already have the proper z-height and we've already collected an image, we don't necessarily need to do that again. So we will deactivate those selections, and we can now add this analysis to our queue. You see that it is at the bottom of our queue list, but we can move it up so it becomes after this survey and before we move to the SiO2. In this case, we'll rename this particular acquisition as PMMA narrow scans, showing how we can adjust the queue on the fly during analysis. Now that our spectrum has continued collecting our survey, we may notice that there are some other peaks that are showing up that were somewhat unexpected. If we zoom in and use our element cursor, we can see these peaks might be something like 10, which could be a contaminant. So going to our PMMA narrow acquisition in our queue, we can go ahead and add 10 to our transition list. So we can collect a spectrum of 10 as well to verify that it is in fact present. In this case, we're not necessarily worried about the chemical state, more about the sensitivity and identification. So we will use a slightly higher pass energy. And we can save that, and that has now updated in our queue for the PMMA narrow scans. So now going back to our full scale of the survey, it looks like we have a fairly good signal to noise, so we can stop the acquisition so we don't need to waste any more time collecting data here. So that file is now saved, and we will move to the next scan, which is our PMMA narrow scan. Now, as you can see, we had 3 transitions we'll be analyzing, and so each of those has its own box for carbon, oxygen, and the tin 3D. Now, within SmartSoft, there are different ways we can display the different tiles, such as the images and the spectra. We can either maximize one particular transition within the bottom section of the screen, as you should see here. We can also maximize the entire display window so we can see things that are more relevant to us as opposed to, say, the intro photo at this time. And we can easily see within just one sweep here that the carbon 1s, we do see multiple chemical states due to the different shifts and shoulders. While the data is collecting, we can do some smoothing of the data, but this will not necessarily be saved with the data file. We can do that in post-processing as well. And we have one more sweep that is being done on the carbon 1s before we go to the oxygen. And again, we do see multiple chemical states, which is indicative of PMMA as we expected. So now we move to the O1s. And here again, based on the PMMA structure, we would expect to see 2 chemical states of oxygen. And that is indeed what it looks like we do see with this scan. And so we will come back and do one more sweep to clean that up a little bit. And then we'll move down to our next scan, which is on the tin 3D. So as we collect the tin, again, we do see the peaks as we thought we saw in the survey. And so we could spend some more time collecting these to get slightly better signal to noise, but we can identify these, and this could be a possible contaminant on our surface. At this time, we can consider this analysis done, and we're now going to move to the SiO2. And so in the same way that we stopped the survey, we can click Stop, which will end the acquisition of the narrow scans, save that data, and now the stage is moving to the SiO2, as you can see on the photo with the crosshair moving to the chosen stage position. And again, this is an SiO2 that we assume to be uniform, and we will do an SXI to confirm that. And as before, we will do the auto Z height alignment and then collect the SXI before our analysis. Again, because this sample is mounted on insulating tape, we will be using the charge neutralization system, which have automatically turned on based on our selection in the queue. And now that the Z height is completed, we will move to the next step, which is our SXI photo, to look at the uniformity of the sample. This is not required, but it is a nice way to look at maybe after the fact if you see something strange in your analysis you can look and see if the analysis was collected in an area where you had some kind of contamination or non-uniformity. And as we can see very quickly and easily in the SXI, there are some dark and bright spots, so it does not look to be a uniform surface. Our analysis is being collected across the center of this image, across the 1.4 millimeter area across the middle of the image. And so if there are some contaminations that are localized, they may get incorporated into our survey, and so it may give us some inaccurate information about the surface. We will spend a little bit more time after this to learn how we can use SXI to identify areas that are uniform and to avoid those or use those for an analysis if we're looking for defects, for example. So in any case, now that the SXI has finished, we'll move to the next step of the queue, which is to collect our survey spectrum. And again, we assume that this is only SiO2 on the surface. And as this first sweep of the survey finishes up, we can see there are several major peaks, likely oxygen at 530, carbon around 285, plus our silicon peaks at 100 and 150. However, it does look like we have a couple of unknown peaks slightly below 500, potentially 10, and around 370, potentially something like silver, which may be contaminants. And we're not sure if those are localized or maybe they are uniform across the surface. And so that would require a little more analysis. We can also use peak ID to identify the peaks. And again, it shows carbon, oxygen, silicon, plus silver. So now that we've seen some possible contaminants in our survey, we can go ahead and stop the survey to save this data. But now we want to do a little more investigation to try to figure out if these dark spots are everywhere on the sample, and if they are they representative of some type of contaminant that we can identify, and then decide where to do our analysis either on or off of the spots. So I'm going to use the SXI imaging, and we can run this similar to like an SEM, in that as we collect these secondary images, we can change the refresh rate of the image. And while I'm navigating, I use the fast mode, so the image refreshes about every 1 second. We can also choose a large field of view while we're navigating, and once we find some features of interest, we can zoom into those and integrate the images to get a cleaner image for our analysis selection. And just like everything else, the SXI is coupled with our charge neutralizers, and so because we have those set to the auto mode, they will turn on as needed for the neutralization during SXI. We'll be using the fast mode, so the image is refreshing about every 1 second at a large field of view, 1400 microns. So I can zoom in on the intro photo to look a little bit closer to where my sample is. And while my SXI is being collected and it's refreshing those images, I can actually click on the SXI itself. And if I click to move to the right, for example, the stage will move, as you can see with the crosshair on my photo, which moves to the right as well. And so there's a correlation between the intro photo and what the SXI and what the X-ray is seeing. So as I continue to click, I can see now that there is a sharp contrast which actually lines up with the edge of my sample. So I can see that to the left is where my sample is and to the right is off of the sample. And so I can continue to basically follow this line by just clicking on the SXI. And if I want to make larger steps, I can go back to the photo and drive directly to that tip of the sample while the SXI is running. And so you can see now we are centered at the tip of the sample in both the SXI and the photo. So now what we'd like to do is maybe go back toward the center of the sample and try to find an area that may or may not have those spots. And we can actually do a small area analysis to determine what they might be. And that may help us determine whether or not there was some type of contamination in my process or the sample handling that now is going to affect my sample surface. So I've seen a slightly dark line in the middle of the SXI, so I can go to total mode, and now the images will integrate on top of each other to give me a slightly sharper image. I can also zoom in to a smaller field of view to really understand the shape of that particular feature. So I'm just going to click and drag a zoom box. And so now I have a field of view of approximately 575 microns, and I'll be collecting images in this integrated mode to see in more detail the contrast area. And so you can see we have kind of a slightly off vertical dark line with a sort of dark spot in the middle of it. And looking at the scale bar of the in the bottom right of the image, this particular spot looks to be about 100 microns in diameter. So what I can do now is I can click and save this particular SXI, and that will add the stage coordinates where this was collected to my position list. And on the SXI itself, I can simply click 2 points for a multi-point analysis. So if I want to do an analysis of the dark spot and then the light area that's more uniform to the outside, I can do that simply by clicking on the SXI. And as I mentioned before, the position of this image is saved in this position list, just like we did for the initial large area surveys. And so what we will do here is the stage is at its current position, and so the X-ray will actually raster and move between these two analysis points without actually moving the stage. And this is a more accurate way of making sure that we're analyzing strictly within those two different regions. Now what I want to do is choose an X-ray size that is appropriate for the feature size. Since the feature is approximately 100 microns in diameter, I'm going to choose a 30 micron diameter X-ray beam, and I can just do surveys like I did before. So I'll start up these surveys again with the auto neutralization turned on. And again, to emphasize, the stage is not actually moving. We are simply rastering the X-ray beam between these two locations. And so shortly we will see two survey spectra being collected, and it will look like that the spectra are being collected simultaneously from these two spots. And it's very close. We choose a analyzer energy window and then move the X-ray beam between those 2 points. And so you have two spectra, a red one and a blue one. The red one is representative of the dark spot, or position -1, area 1, and the blue spectrum is representative of the more uniform gray area 2. So far they look fairly similar, but now that we get to the oxygen peak around 530, we can see that there is a little bit of difference in the intensity between these two locations. The blue spectrum, which is on the uniform gray area, is higher in intensity than the red, which is on the dark spot. So it looks like the uniform gray area has more oxygen than that dark spot does. So now we can go back to our full scale and we see a few more peaks, such as the carbon at 285, and it looks like we also do see a difference in the intensity at this region as well. In this case, it looks like the red spectrum, or area 1, the dark spot, does have higher intensity. So potentially what we could be seeing is some sort of carbon contamination spot on the surface. Now one thing we also notice while we're in this same view is that it looks like we do see in the red spectrum that maybe there's an additional peak that could be calcium, which could be a localized contaminant because we did not see it on the blue spectrum. We can also toggle between these two spectra and do peak ID to look at the two locations. We can do automatic peak ID, or we can label manually with our periodic table. So if I label the calcium, it does look like maybe there is calcium in that spectrum, But what we can do as before is we can import the transitions that are labeled on the spectrum and then set up a narrow scan to confirm that. Now that we have run a couple sweeps of the survey, the next step is to do a high resolution analysis, in this case of the calcium, just to confirm that it is in one location and not the other. So for our narrow scans, we tend to use lower pass energies. So in this case, we'll use 55 for the carbon, oxygen, and silicon, and for the calcium, because we're more or less trying to confirm its presence as opposed to the chemistry at this stage, we will set it to a higher pass energy. I can also reorder the acquisition. So in this case, the calcium is the one I'm most interested in, so I can set that to be run first. I can then add that to my acquisition queue. And just like before, we can stop the currently running acquisition because we feel like we have good enough data to move to the next step. So it will finish the currently running sweep and then move to the next step, which is our calcium high resolution scan. And just as before, the stage will not be moving. We will be rastering the X-ray beam between location 1, the dark spot, and location 2, which is the uniform gray area, to the bottom right from there. And we'll start by looking at the calcium to confirm whether or not it is indeed on that dark spot. And so it appears that we do see a calcium peak on the red spectrum, which is our dark spot. And if we look at each of the spectra individually, we can see that yes, it looks like it is on the red spectrum, but not necessarily on the blue spectrum, confirming that it appears to be a localized contaminant in this case. And this was all done by using our SXI imaging to identify defects quickly and then use multipoint analysis to confirm their presence. So in conclusion, with this analysis, we have performed both large area and small area acquisitions on insulating samples using automated processes that allow us to turn on and off our charge neutralization system, do automatic Z-height alignment, collect an SXI, which allows us to look at the uniformity of the sample even before we collect our data, and then use SXI again to identify areas that might be contaminants and choose multiple points for analysis to confirm whether or not they are localized.